Hello everyone. I am pleased to share our study on the gut microbiome and lipid metabolism in early pregnancy. We aim to identify molecular changes that precede the diagnosis of gestational diabetes mellitus and to examine how the gut microbiome relates to host lipid metabolism. Gestational diabetes is a common pregnancy complication that affects both mothers and children. It increases women's later risk of type 2 diabetes, while their children are more prone to obesity and glucose dysregulation. Diagnosis usually occurs at 24 to 28 weeks, although biological changes may begin earlier. We therefore focused on the first trimester and combined population multiomics with laboratory experiments. Our study had 4 main findings. Microbial and lipid changes were already present in the first trimester. Specific microbes may contribute to metabolic dysregulation through lipid metabolism. Experimental findings supported a role for glycosphingolipids in microbial regulation of insulin sensitivity. Finally, microbial and lipid information improved early risk stratification. This study included 784 women from the Tongji-Huaxi-Shuangliu Birth Cohort. All provided stool and fasting blood samples in early pregnancy and underwent screening in mid-pregnancy. During follow-up, 222 developed gestational diabetes and 562 maintained normal glucose levels. We obtained gut metagenomes from all 784 women, plasma lipid profiles from 738, and genetic data from 689 participants. Women who later developed gestational diabetes had lower Xiaonan and Gini-Simpson indices in early pregnancy, indicating reduced microbial diversity. Their overall microbial community structure was also different. These alterations therefore preceded the clinical diagnosis of gestational diabetes. At the species level, 26 microbes were associated with later gestational diabetes. One was Ruminococcus bicirculans. This bacterium was less abundant in women who developed the condition, and higher abundance was associated with lower risk. 7 species showed consistent associations in external type 2 diabetes datasets. We also identified 22 associated microbial pathways. Fatty acid and lipid biosynthesis pathways were enriched, whereas pathways related to fermentation, carbohydrate metabolism, and nucleotide metabolism were depleted. Plasma lipid profiles also differed before diagnosis. We identified 165 associated lipids across 17 classes or subclasses. Triglycerides, diacylglycerides, and some lysophosphatidylcholins were generally positively associated with gestational diabetes, whereas several phosphatidylcholins and sphingomyelins were inversely associated. Cross-omics analysis identified 970 associations between microbial and lipid features. Exploratory mediation analysis suggested that lipid variation could statistically account for part of the association between the microbiome and gestational diabetes. Dihexosylceramide-24:1 was the most prominent lipid feature and accounted for 9.6% of the association between Ruminococcus bicirculans and gestational diabetes. This lipid may contain several structural isomers. We therefore selected lactosylceramide-24:1 as a biologically plausible candidate for experiments. Under anaerobic conditions, Ruminococcus bicirculans was cultured with glucosylceramide-24:1 and galactose. After 48 hours, lactosylceramide-24:1 was detected, and its production increased with substrate concentration. This showed that the bacterium can produce this lipid in culture. We next examined whether this finding was relevant to glucose metabolism. In a diabetes model induced by a high-fat diet and streptozotocin, gut microbes were reduced with antibiotics before bacterial colonization or lipid administration. Both Ruminococcus bicirculans and lactosylceramide-24:1 improved insulin tolerance, as shown by lower glucose levels and a smaller area under the curve. In primary hepatocytes, lactosylceramide-24:1 enhanced insulin-stimulated phosphorylation of protein kinase B at threonine 308 and serine 473, without changing total protein kinase B. These experiments suggest that the bacterium may influence insulin signaling and glucose regulation through glycosphingolipid metabolism. Because clinical factors alone have limited ability to identify high-risk women in early pregnancy, we tested whether multiomics data could improve risk stratification. The area under the receiver operating characteristic curve was 0.80 for the lipidome, 0.68 for the microbiome, and 0.56 for clinical factors. Combining the lipidome and microbiome increased the value to 0.82, while the fully integrated model reached 0.84. We also constructed microbial, lipid, and genetic risk scores. Compared with women with low microbial and lipid scores, those with high scores in both had an odds ratio of 19.19 for gestational diabetes, together with a significant additive interaction. In summary, changes in the gut microbiome and circulating lipidome related to gestational diabetes were already present in early pregnancy. Population multiomics and experimental findings suggest that Ruminococcus bicirculans may influence insulin signaling and glucose metabolism through glycosphingolipid-related processes. Microbiome and lipidome data may also improve early risk stratification. Further validation in diverse populations and pregnancy-relevant models is needed. Thank you for watching.